Maybe 100 points or something like that for extra credit? Okay. The delta G problem was worth 20 points. That's right. So thanks for that feedback. So let me ask you a question. So one comment, I've heard this from others as well. They felt that the delta G problem should be worth less points. Okay, okay. All right. So if I do less points, that means I've got to fill it in with another question. So if I had another long problem, do you want that on the exam? How many want another problem in place of a 20-point? How many want to have the 20-point delta G with more time? See, it's about split. So, and I'm sympathetic to that. I understand that. One of the issues I always deal with in writing an exam is the more problems I put in, the less each question is worth. And people tend to like that. But then they say, I'm out of time. And so I've got to try to balance, obviously, time with the performance on the exam. Time with the covering the material on the exam. Performance obviously comes in as another matter. But that's kind of why I have that. So I felt that the delta G, because it is an important concept, really needed to be worth as much as it was. But I do hear your comments on that, and I do appreciate that. Other comments? Everybody's quiet. Okay. All right. So the exams are being graded. The TAs had a massive exam of their own today, so they haven't worked on them over the weekend. They will work on them very hard. I told them I wanted to have them back to you, if possible, by Wednesday. So the aim, and don't quote me on this because they can only do, they're only human, they can only do what they can. We will make every effort possible to get them back to you by Wednesday. When they are available, I'll put a notice, an email out to you, as I've done before, saying the exams are available, et cetera, et cetera. And please don't bug people in the office until you get that email from me. Okay? And as I said, hopefully we'll have that for you on Wednesday. All right. Well, we're actually ahead of where we need to be, so I'm feeling pretty good about that, except for the fact I'm catching a cold, and which is why I may finish early today, so I save my voice. Of course, if I completely lost my voice, then we wouldn't have class at all, so maybe that would be even better. Who knows? All right. So we've been talking about glycolysis, and last time I hope I made a case for you that high fructose corn syrup has some problems. Okay? And it's not completely understood about why that is a problem, but it's certainly a phenomenon. And it's not totally coincidental, I think, that the American obesity epidemic has paralleled the use of high fructose corn syrup in soft drinks and other foods. Excuse me. So what I want to talk about today is mostly related to regulation. And I'll say a little bit about gluconeogenesis. We'll get started on that. I'm not going to finish gluconeogenesis today, obviously. And that's not even on tap for today's lecture as it stands anyway. So, okay. Well, last time I talked about the high fructose corn syrup. The one thing in that section I didn't talk about was a common topic that comes up. Several of you have asked me about this, and this is lactose intolerance. So lactose intolerance is, as I pointed out last time, not related to lactate, which is a different molecule, but lactose is a disaccharide, and breakdown of lactose is a problem for some people. The breakdown of lactose into galactose and glucose is catalyzed by the enzyme known as lactase. Okay? There appears to be some differences ethnically as well as, not totally ethnic as well, with respect to deficiency of lactase in adulthood. Now, if you think about it from an evolutionary standpoint, it sort of makes sense that we wouldn't be making lactase as adults. We're not typically nursing when we are beyond a year or two old, and so the need for that enzyme really, in an evolutionary sense, shouldn't even be there. So it's not surprising that many adults don't make this enzyme because milk was not a part of our diet as we were going through the evolutionary changes. The deficiency of this enzyme results in some serious problems relating to digestive pain. The digestive pain arises from the fact that lactase is normally abundant early in the digestive process, And at that point, it can break down galactose and glucose, which then get absorbed into our bloodstream and provide energy for us. If lactose is not broken down early, it passes through the place where it would be easily absorbed, and the bacteria that are in the gut have a heyday with it. And so they take that lactose and break it down into whatever it is that they happen to be able to metabolize. E. coli can, for example, break down lactose. And the byproduct of that action by them is the production of a tremendous amount of gas. So people who have lactose intolerance have, as I say, abdominal pain. They have a lot of gas, etc. And it's because the bacteria are breaking down the lactose and using it instead of you breaking down the sugar and using it. Now, this can be fairly easily treated. There are tablets that you can take that contain the enzyme lactase and will, in fact, help to break down lactose if that is a problem for you. So it's not a severe problem, but it certainly can be one that causes some discomfort. Okay, let's see. So it's time we spent some time now talking about regulation, because I've said a little bit about it, and I've told you that there are 3 enzymes in glycolysis that are, in fact, regulated. The 3 enzymes, Gesundheit, I'm sympathetic to that. The 3 enzymes are hexokinase, about which I won't say very much at all. The second enzyme is phosphofructokinase, or PFK. And the third enzyme is pyruvate kinase. So I'll say more about PFK and pyruvate kinase than I will about hexokinase. Now, even though I said I wasn't going to say much about hexokinase, I will just briefly tell you why I'm not going to say much about it, and then we'll move on. Hexokinase has an unusual regulatory scheme called substrate regulation, and it's the availability of substrate and plus the Km value of hexokinase that helps to regulate that enzyme. So it can and does play a role in the breakdown of glucose. From our purposes, we don't need to get into the kinetics of it, so I don't really spend much time talking about hexokinase. I do want you to know, however, that it is an important enzyme regulating the overall pathway. As I said earlier, the most important enzyme with regulating the pathway is PFK. PFK, it turns out, is allosterically controlled. Stupid thing. There. The PFK is allosterically controlled by several compounds, as we shall see. And one of the things you start to see with allosteric regulation is that cells actually are using important indicator molecules to determine whether or not an enzyme is active or inactive, or more active or less active, as the case might be. This figure shows an interesting sort of thing. And at first glance, it looks like it makes perfect sense. Okay? When we have PFK and it's catalyzing a reaction, and we have low amounts of ATP present, we see a greater velocity than when we have high levels of ATP present. So first of all, what does that tell you? ATP is somehow allosterically inhibiting the enzyme, right? Okay, so at first glance, that makes sense. We've seen this before with ATCase. With ATCase, we add CTP. We see something very similar to this. There's a less activity when CTP is present. But there's something very confusing about this observation right here. The confusing thing is that the reaction that phosphofructokinase catalyzes requires the following things. Fructose 6-phosphate plus ATP. That gives fructose 1,6-bisphosphate plus ADP. Now, how in the world can a lower amount of ATP give more activity than a higher amount of ATP, which is what this figure is telling us? ATP is a substrate for this reaction. You've never seen this before. It's pretty unusual. How can we explain this? Everything we've seen so far about velocity and substrate concentration says that the greater the substrate concentration, the greater the velocity. What's going on here? Yeah. Is what? Well, feedback inhibition is a phenomenon where the end product of a pathway inhibits an enzyme early in the pathway. This is definitely not feedback inhibition. It's an inhibition, though, definitely. It's not Le Chatelier's principle. Le Chatelier's principle would say the more of this stuff we had, the faster we should go. There's something else going on here. It's a trick question. Okay? Think outside the box here. That's a hint. Yeah?
Is it allosterically inhibiting and being used as a substrate?
It is allosterically inhibited and being used as a substrate. But how's it doing that? Yeah? Nope. The only concentration that matters is ATP. Yeah, back in the back, yeah. Nice try, but no, it's not turning on something else that affects the enzyme. Yes, sir?
Does that do with the fact that at the end you produce a lot of ATP, but at the end of the whole process?
At the end of the process, you produce more ATP. It's a nice try also, but unfortunately, no. Is it because it's a reversible process? Nope. Yep. I see, the more I get you guys talking, the less I have to talk. Yeah.
Maybe the active site has a higher affinity for ATP than the allosteric.
Aha! He hit it. There's two ATP binding sites. One that is the active site, and the active site has a different affinity than the allosteric site. So there's two different places where this enzyme could bind ATP. My question to you is, which one has the lower Km? Km, you remember, we talked about that. Measure of the affinity of an enzyme for its substrate. The lower the Km, the higher the affinity. Which one is going to have a higher affinity for the substrate? The active site or the allosteric site? The active site. That explains this phenomenon. All right? In the presence of small amounts of ATP, the active site binds the ATP. The allosteric site doesn't even have a chance. When ATP concentrations get high, it starts loading into the allosteric site and turns the enzyme off. It's a very cool system. So only in this way can a substrate be an allosteric effector as well and turn an enzyme off, or turn it down, as you can see here. So that's what's actually going on with this enzyme. All right, so what that means is important, because if ATP levels are high, do we want to be breaking down glucose? No. It's kind of like lighting a fire in your fireplace when it's a summer night. It doesn't make any sense. You're wasting energy if you're doing that. And so we don't want to be breaking down glucose if ATP levels are high. So the level of ATP helps tell the cell, it helps tell the cell, shut this process off. We don't need any more energy. Let's not burn up our resources. Let's save our fuel for when we need it later on. Okay, now, here's another interesting observation. And this interesting observation concerns another molecule. The other molecule is called fructose 2,6-bisphosphate. You can call it F26BP. Now, F26BP is not, underlying not, a substrate in glycolysis. It sounds like it, but it's obviously different from the one that is in glycolysis, which is F16BP. So fructose 2,6-bisphosphate affects PFK. How does it affect it? Positively or negatively? Based on what you see in the plot, how does it affect it? Positively or negatively? It's positively, right? We see more velocity when we have F26BP present than when we have F26BP absent. Okay? So that tells us that F26BP is an allosteric activator of this enzyme. F26BP is a very interesting compound. It was only discovered in about the 1980s. And the reason it was only discovered then was its concentration in cells was so low, it was almost impossible to detect it. What that means is this enzyme is exquisitely sensitive to F26BP. It doesn't take much F26BP to actually activate this enzyme by a considerable amount. This activation occurs in spite of increased concentration of ATP. So F26BP can kind of overrule, as it were, ATP in the allosteric regulation department. Here we see the F26BP, okay, the reaction in the presence of F26BP and in varying amounts of ATP, and we see even so it's way up here compared to the reactions without that. So this is a very, very sensitive activator of phosphofructokinase. Now keep in mind, as I said, this molecule is not, underline not, a substrate in glycolysis. This will confuse some of you later when we get talking about some of these things. Okay? Well, how do we make F26BP? It turns out that we make F26BP from fructose 6-phosphate. This shows it as if it's a part of glycolysis. It's not. It's just a separate enzyme that makes F26BP from F6P. That's all that it means. All right? So F26BP is not a part of the glycolytic pathway. It's just simply another molecule that regulates PFK. Okay? When PFK is activated, now this process goes gangbusters and moves everything forward. The structure of fructose 2,6-bisphosphate looks like this, as you might imagine. Remember drawing our sugars, we always number them, so we have carbon number 1, 2, 3, 4, 5, and 6. There's carbon number 2 right there. It's coming off of carbon number 2, and that's what it looks like. Okay. Let's not talk about that. And let's not talk about that. Let us turn our attention briefly to discuss the regulation of pyruvate kinase. Is there any question about that F26BP before I go forward? Yes?
I don't understand, but where does it bind to the PFK?
It binds on the PFK at the end. It has an allosteric binding site for it. Yes, sir?
Does it just stay in the system, or does it get broken down back?
So it's a good question. What happens to F26BP? All right, does it just stay in the system? Does it get broken down, etc.? It does get broken down. In fact, what we will see when I talk about gluconeogenesis is that the breakdown of F26BP and the synthesis of F26BP are very tightly controlled. And as you might imagine, this is a fairly important thing to do. We only want to turn on glycolysis when we need energy. We want to turn off glycolysis when we don't need energy. And so controlling this through controlling the levels of F26BP is also very important. Now, actually, before I just remember one other thing I wanted to say before I talked about pyruvate kinase. There are other allosteric effectors of PFK that aren't shown here. I'll talk about them later. But I will tell you about one of them briefly here. And this is, again, talking about PFK still. This other regulator is interesting, and I think if once I tell you what it is, you can figure out whether it's an activator or an inactivator of the enzyme. Okay? The other effector that affects PFK is AMP, adenosine monophosphate. Not cyclic AMP, AMP. What does AMP indicate inside of cells? Low energy. So in low energy, do we want PFK going or not? Yes. So AMP is an allosteric activator of PFK. Okay, and as I said, I'll talk more about that. What happens is once I talk about gluconeogenesis, I talk about the regulation of the two pathways together because one affects the other one significantly. Okay. All right, pyruvate kinase. So the last enzyme of glycolysis is pyruvate kinase. Pyruvate kinase is regulated both by allosteric effectors and also by phosphorylation. Okay? Phosphorylation. So we see that in the dephosphorylated state, pyruvate kinase is the most active. Okay? In the phosphorylated state, that is, if I put a phosphate onto it, it is less active. How does that phosphate get onto there? Well, this is the first place you're going to see it. You're going to see this increasingly over the next couple of weeks, and that is protein kinase A. How do we activate protein kinase A? That was the epinephrine pathway, right? Epinephrine pathway led to the production of cyclic AMP, which activated protein kinase A. And protein kinase A, one of the things it does is it will phosphorylate protein kinase. It will phosphorylate pyruvate kinase. All right. Now, this means it's less active when the phosphate is on there. Does it mean it's inactive? No. Okay. It's not unlike what we talked about before with turning the volume up and turning the volume down. It's less active when the phosphate's on there. The enzyme is most active when it is dephosphorylated and when F16BP is present. Now, that was part of that scheme I described to you earlier, which I called feed-forward activation. Remember that? Remember that? Okay. I did talk about that, right? All right. So feed-forward activation happens when F16BP accumulates. And why does it accumulate? What reaction, what enzyme catalyzes a reaction where F16BP accumulates? Aldolase, right? That aldolase reaction has a high positive delta G0 prime. That means that if nothing else happens, F16BP will start to increase. As it starts to increase, it can affect pyruvate kinase. It binds to pyruvate kinase and starts this process, which means that we reduce the concentration of phosphoenol pyruvate, which means we reduce the concentration of 2-phosphoglycerate, which means we reduce the concentration of 3-phosphoglycerate, which means we reduce the concentration of 1,3-bisphosphoglycerate, which means we reduce the concentration of glyceraldehyde 3-phosphate and DHAP, and those are the products of the aldolase reaction. So we're pulling that aldolase reaction by this feed-forward activation. So F16BP and dephosphorylated pyruvate kinase create a very active enzyme. On the other hand, if we want to inactivate the enzyme, two things combine to help make it inactive. One is phosphorylation. The other is ATP. Now, this sort of makes sense as well. If we have a lot of ATP, do we want to be running this reaction? No, we don't. Again, we don't want to be burning gasoline in our fireplace if it's a summer night. We don't need that extra heat. We're going to be wasting energy if we do that. Okay? Now, there's another place where we have high levels of ATP. And the other place where we have high levels of ATP are when we're synthesizing glucose. When we have a lot of ATP laying around, we don't want to just have it sitting there doing nothing. We want to have it charging the batteries, as it were. That is, storing that energy in some chemical form. ATP is not a good long-term storage form of energy. So we want to take that ATP and convert it into something else. So one of the something else's we will convert it into is glucose. The synthesis of glucose involves a reversal of the glycolytic pathway. It's not an exact reversal, as we shall see, but it's a reversal of the glycolytic pathway. Imagine, if you will, that we start, and in fact, I'll just briefly tell you, when we start with pyruvate, we can't just directly reverse this reaction. This was the Big Bang reaction. As soon as I make, back up. If I try to reverse this reaction, I can't, because there's so much energy given off in this reaction, it's almost impossible to make anything here. This was the reaction, as you recall, it created enough energy almost to make 2 ATPs. The concentrations necessary to make this reaction go backwards are impractical inside of cells. They're impractical. Okay? So cells have to do a two-step. They have to do 2 reactions. They convert pyruvate to oxaloacetate and oxaloacetate back to PEP. We'll talk about that in a bit, so you don't need to write that down at the moment. But let's imagine that the cell has decided it's going to make glucose. I've got all this ATP sitting around. I do my 2-step. First step, oxaloacetate. Second step, phosphoenol pyruvate. What happens if I have not inactivated this enzyme? It's going to go right back to the right. I will just have completed a triangle. It's something that's known as a futile cycle. Cells don't want to go through futile cycles, because if they do, all they do is waste energy and produce nothing. Okay? So it's important when the synthesis of glucose is going on that this enzyme be turned off. And that's why cells regulate the last enzyme in this pathway. It's unusual, but that's the reason why they do it. Do you have a question?
Yeah. I mean, when you have PK active, it's when you have lots of epinephrine, that's a flight or fight sort of thing. Why would your, I mean, I understand maybe your liver, but why would your muscle cells want to decrease your glucose metabolism in a fight or flight?
Okay, so his question is, let me come back to the answer to that later, but I'll just briefly tell you what his question is so that you can understand what his thinking is, and we'll see why this happens later. His question is that epinephrine is adrenaline, fight or flight, right? I have to decide to run, get the heck out of there. So why do I want to be stopping my glycolysis process if I'm doing that, okay? And the answer to the question briefly, and we'll see that why this happens later, is this happens primarily in the liver. It's not happening in the muscle, because in the muscle, you're exactly right, we don't want to be stopping this process from going because we want to have as much energy as we possibly can. Okay? Make sense? And you might wonder how that's controlled that way, and the way it's controlled is epinephrine doesn't affect your muscle cells. It affects your liver cells. Yeah. But good, very good question. Okay, well, as I said, we'll talk more about that. All right. So regulating pyruvate kinase is very, very important consideration when we are talking about making glucose. Now, one of the things I've already talked about briefly prior to this were the glut proteins. The one you specifically saw was GLUT4. GLUT4 was this protein that was brought to the cell membrane by stimulation of cells with insulin. That brought the GLUT4 protein to the cell surface, and when it got to the cell surface, it brought in glucose. So the function of all of these GLUT proteins is to bring glucose into cells. Okay? We see a variety of GLUT proteins. We have a glut of GLUT proteins, as it were. Excuse me. And they have special places where they're located, etc. We're not really focused on that as much as understanding what they do. Now, these GLUT proteins become important when I talk about the phenomenon of cancer. Okay? Now, cancer cells are, as you know, different from other cells in a few senses. They are, in fact, the origins of cancer cells are regular cells. But cancer cells typically will have, for example, a division rate that is faster than the cells around them. That's how a tumor forms. We get this sort of growth, this uncontrolled growth and mass of cells that arise to form a tumor. There's several steps that's involved in making a tumor, and we don't know all of the steps. We know some of the steps. We don't know all the steps that's involved in taking and making a cell into a tumor. But we do know this, that one of the critical things that has to happen during the tumorigenesis process, that is the process of forming a tumor, is that the tumor needs oxygen. The tumor needs oxygen. Just like all the rest of your cells need oxygen, the tumor needs oxygen. Why? Well, if a tumor cell doesn't have oxygen, what's going to happen to its synthesis of ATP? It's going to fall, right? When it falls and it doesn't have oxygen, what does it make? It makes lactate. It goes through fermentation, right? So if I look at a tumor cell, and by the way, tumor cells, especially when they're starting, are fairly deficient in oxygen. They're fairly deficient in oxygen. One of the strategies for killing tumor cells is to destroy their oxygen supply. Okay? Destroy their oxygen supply. All right? There are proteins that are involved in making blood vessels that will innervate into your body. They're present all over. Your body's making these hormones that tell the body, okay, make some more blood vessels that deliver blood to these various tissues. The tumor cell will do that as well. So one of the strategies for killing tumor cells is to inhibit the action of those hormones that stimulate the growth of blood cells and literally starve the tumor to death. Okay? And in some types of tumor, those types of inhibitors appear to work very well. Now, I'm not going to take that approach here in what I'm going to tell you, but I think you'll see why I'm talking about it right now relative to glycolysis. Remember that tumor cells are generally starved for oxygen. That's an important consideration. This shows a mass of a tumor. Okay? And we can see that if blood vessels get a chance to grow in there, they will. And these tumor cells make this hormone that says grow blood vessels here. Okay? If they don't make that, they probably never make it to the tumor stage. You probably never know about them. You're probably making tumor cells right now, and if they're not making this hormone that stimulates blood vessel growth, they are not, in fact, probably going to make it as a tumor cell. They'll probably die. Well, before they get to that stage, any cell in your body has a recognition that its oxygen concentrations are low. Your muscle cells have this, your liver cells have this, your brain cells have this. They all have the recognition that oxygen concentration is low. When oxygen concentration is low, we have a phenomenon known as hypoxia. My battery is burned out here. Okay, so hypoxia refers to the fact that the cell is existing under low oxygen concentrations. The cell has a way of dealing with those low oxygen concentrations, at least over a short term. One of the ways it deals with it is it synthesizes a protein called HIF1. HIF1 stands for hypoxia induction factor. So when a cell is short of oxygen, it will make HIF1. I'll tell you what HIF1 does in a minute, but you need to know that when oxygen is low, HIF1 is produced. Induction. Now, what happens then, all right, is HIF1 is a transcription factor. Anybody know what a transcription factor is? Okay, so transcription factors are proteins that bind to DNA and help control transcription of specific genes. If we're going to make various enzymes, various proteins, we have to first make the messenger RNA for them. That's transcription. And HIF1 controls the transcription of several genes. Now we've got the stage set to see what this guy actually does, and I think it's pretty phenomenal. Here are the proteins, all right, in glucose metabolism that are activated by HIF1. 2-gluts, Hexokinase, glycolysis enzyme, PFK, glycolysis enzyme, aldolase, glycerol 3-phosphate dehydrogenase, phosphoglycerate kinase, enolase, pyruvate kinase. That's 1, 2, 3, 4, 5, 6, 7 of the 10 enzymes of glycolysis are turned on by this enzyme, by this transcription factor, HIF1. Okay. Now you might wonder, well, why does it turn on the enzymes of glycolysis? That'd be a real good exam question. Why the enzymes of glycolysis? What is increasing the amount of enzymes of glycolysis due to this process? Okay, so he's exactly right over here. All right, cells, when they're low on oxygen, can't do aerobic metabolism. They need anaerobic metabolism. All right. In anaerobic metabolism, we decided we got 2 ATPs for every glucose. Right. When we had aerobic metabolism, we had 38 ATPs per glucose. Right. That means, therefore, that for a cell to have the same amount of energy, it needs 19 times as much glucose. Well, you start dumping 19 times as much glucose into a cell, you're going to need more enzymes to handle it. So, HIF1 induces the production of these enzymes of glycolysis, and that helps keep that low oxygen cell alive. That makes sense? Yes, Shannon.
Where does all the glucose come from?
Where does all the glucose come from? Blood supply. Blood supply. They just start sucking in all the glucose out of the blood supply. Yep. And the glucose, the bloodstream has a pretty constant supply of glucose. When something is really sucking the glucose out of it, the liver starts producing more. So it's a very steady pipeline of glucose for the most part, unless you're starving yourself or something. Okay, that makes sense to everybody? Good. Okay, so I've decided what I'm going to do is I'm going to introduce the topic of gluconeogenesis, and then we'll finish early, since my throat would appreciate that. All right, any other questions about this before I go and do that? Okay. All right, well, we start glucose synthesis talking about something kind of odd, and that's sorry, that's glycerol metabolism. Why in the world do we talk about glycerol metabolism? Well, the reason that we do is we're interested in making glucose. And glycerol is produced in our body as a product of the breakdown of fat. Okay? The fatty acids that are produced by the breakdown of fat, fat is comprised of fatty acids linked to glycerol. The fatty acids are worthless for making glucose. We can't convert the carbons in fatty acids, for the most part, into glucose. The only part of the fat molecule that we can convert into glucose is glycerol. And we do it by this scheme. Glycerol is converted to glycerol phosphate, which is in turn converted to dihydroxyacetone phosphate. And dihydroxyacetone phosphate, being an intermediate in glycolysis, can go backwards up the cycle and make glucose. Okay, so that's actually it's a piece of trivia is what it is. That's the only part of a fat molecule that can be made into glucose. All right, now let's spend a little bit of time, and then we'll finish for the day, thinking about the process of the synthesis of glucose, starting with simple precursors. All right, simple precursors. We said the end product of glycolysis was pyruvate. Okay, the end product of glycolysis was pyruvate. We are going to call the start of the process of synthesizing glucose as pyruvate as well. Well, the process of synthesizing glucose is called gluconeogenesis. And in this process, pyruvate is converted. If we went down for breakdown of glucose, we're going to go up for the synthesis of glucose. And it's here that we see the reversibility of reactions is a very important consideration. 7 of the 10 reactions of glycolysis are simply reversed in gluconeogenesis. That is, they run the other direction. In order to run the other direction, what do I have to do for any reaction? Let's say I've got A going to B. If I want to make it going backwards, what do I have to do? I have to increase the concentration of B, right? So if I want to go up this pathway, I darn sure better have a lot of starting material to go up the pathway. Pyruvate turns out to be a compound that we have in pretty good abundance. For one, we can convert the amino acid alanine into pyruvate very easily. So if I'm on a protein-rich diet, my body can still make glucose because it can make pyruvate very easily. All right, so I'm going to briefly step you through a couple of reactions, and then we'll quit for the day. The first reaction, as I said earlier in the lecture today, is that pyruvate cannot be converted directly back to PEP. That would require reversing the pyruvate kinase reaction that is almost impossible to reverse because the delta G 0' is so negative in the forward direction. We have to do what I call a two-step around it. So the two-step involves 2 additional enzymes that are in gluconeogenesis that were not in glycolysis. In fact, if you look on this scheme, you'll see the enzymes that are unique to gluconeogenesis labeled in red. And I'll point out to you that they're right here where pyruvate kinase was. That was a regulatory enzyme. There's one up here where PFK was. That was a regulatory enzyme. And there's one up here where hexokinase was. That was a regulatory enzyme. So the 3 regulatory enzymes of glycolysis are bypassed with different reactions in gluconeogenesis. I'm only going to talk about these 2 down here today. All right, so let's say that I've got pyruvate sitting here. And my body has plenty of ATP. I've been sitting around and not exercising as I should be, and therefore I have plenty of ATP, I have plenty of pyruvate. To reverse this process, an enzyme called pyruvate carboxylase is necessary to start the overall gluconeogenic pathway. Pyruvate carboxylase is an enzyme that is found only in the mitochondria. It's not found in the cytoplasm. All of the glycolysis reactions occurred in the cytoplasm. So now for the first time we're seeing sequestering of an enzyme, and the enzyme that's sequestered is pyruvate carboxylase. It's found only in the mitochondria. Okay? This is a reaction that requires some energy. To go from pyruvate to oxaloacetate, which is the product of that reaction, requires a CO2 because we're going from a 3-carbon molecule to a 4-carbon molecule. Oxaloacetate has 4 carbons, pyruvate only had 3. So we're adding a carboxyl group. It requires ATP energy. And keep in mind that since we're going to make a glucose, we're going to need 2 pyruvates to ultimately make a glucose because we need 2 times 3 carbons to get there. 2 pyruvates plus 2 ATPs plus 2 bicarbonates gives us 2 oxaloacetates. Now you'll notice over here that lactate and some amino acids, in this case alanine, can give us pyruvate. Some amino acids, like aspartic acid, can give us oxaloacetate. So we don't even technically have to start at pyruvate in order to go up the pathway. Oxaloacetate has 4 carbons. Excuse me. We need to get back to 3 carbons, and that involves now a decarboxylation going up the pathway. It requires another enzyme, and that other enzyme is called it's called phosphoenolpyruvate carboxykinase. Mouthful of a name. You can call it PEPCK. PEPCK. Not Pepsi Cola, PEPCK. PEPCK. PEPCK is found only in the cytoplasm. Only in the cytoplasm. That means that that oxaloacetate that we made had to be moved out of the mitochondria into the cytoplasm so that the next reaction can proceed. Once we're at PEPCK, everything else is going to be a reversal of glycolysis until we get up here. So PEPCK goes to 2-phosphoglycerate, goes to 3-phosphoglycerate, goes to 1,3-bisphosphoglycerate, goes to, remember we had 2 of these guys, so we have 1 goes to glyceraldehyde 3-phosphate, 1 goes to dihydroxyacetone phosphate. These 2 guys combine to make fructose 1,6-bisphosphate, and then we're at the point where we have another enzyme. That is a good place to stop. Oh, by the way, before you disappear, I mentioned last week if you want to come over and have Thanksgiving dinner with me, I'd like to invite you. I need to know if you're going to come, though. So please let me know if you'd like to come over to my house and have Thanksgiving dinner.
So, I want to go.
Oh, okay. What's your name?
Kelsey.
Okay. Let me give you my address.
I got stuck in town. My parents live in Seattle.
Oh, good. That's one of the reasons we have it, is so people can have that aren't going somewhere else can come. It's always fun for us. So our address is 3640 Northwest Harrison. And we'll probably have dinner about 4. I say come over 2:33. We'll sit and chat and so forth before dinner. Okay?
All right, awesome.
Happy to have you. Take care. Hi.
So, we're going to at 10 tomorrow. Does that still work?
If I said it did, it does. Did we get it set up? Yes. Okay. I can't remember. I don't have my calendar in my head. Hi.
Can I bring my 5-year-old?
Sure, sure. Yeah. So let me give you our address. Let me get this turned off here first. Yeah, be most welcome. That'd be cool. So, how you doing, Emily? Good. I looked at the videos. I don't think we're going to use them.
Really?
They didn't turn out very good. No.
It's a couple of us, so it's kind of
it's just too weak. There's not enough voice there, so I haven't put it out yet. But I did look at them. And I thought, oh, that's too bad. So we're at 3640 Northwest Harrison, and we're going to eat about 4 o'clock. So I'd say come over about 2:30 or 3, and we'll sit and talk and so forth. Get to know each other a little bit. Okay. And your name is
Chitra.
Chitra. That's right, Chitra. Okay, sir.
Is there a good time to come by your office today? First of all, I had one question, and then you said
Come on by. I can't remember what my schedule is, but I'll squeeze you in. Okay.
I'm free until like 5, so what
Okay.
time would be good?
Look at my calendar. I honestly don't remember off the top of my head, but come by. I'll be there. I'll be there. Hi. You're coming over. That's right. You have our address.
Yes.
Okay. So
They're walking on the or
so you
working on the
know where we live.
Yeah.
Okay. Good.
But do you guys want anything?
No, just come on over. We'll eat about.
My mom can make something really good.
That's fine. We will eat about 4. So come on over about 2:30 or 3, and we'll sit and talk and have a good time.
Nice.
You join us?
I can't remind you, James.
Okay. Take care.
Question, because I was curious.
Sure.
So